Hey everybody. So up to this point, we've been talking about nutrient acquisition and photoautotrophs. We're going to switch gears now and talk about nutrient acquisition in the chemoheterotrophs, and we'll be talking about fungi and animals. And just remember that the chemoheterotrophs acquire their energy from chemical compounds as opposed to sunlight, and they get their carbon from organic compounds. Okay, so that's as opposed to inorganic compounds like CO2. Organic compounds are organic molecules. In other words, they eat other organisms as a carbon source. And so first we're going to talk about fungi. But before we get into fungi, I want to talk about the two general methods that chemoheterotrophs use to acquire reduced carbon and other nutrients. And remember, when I talk about reduced carbon, what I mean are high energy carbon molecules. So something like glucose would be an example of a reduced carbon molecule. And so there's two major strategies or general methods. One is absorptive heterotrophy. The other is ingestive heterotrophy. So absorptive heterotrophy, that's a strategy that's used by fungi and chemoheterotrophic prokaryotes. And essentially what absorptive heterotrophy is absorbing small molecules that are present in the environment, and this means molecules that are small enough or tiny enough to get through a cell membrane on their own, or first secreting some enzymes so that they can break down larger molecules into molecules small enough to move across the cell membrane through a process of extracellular digestion. So remember, extracellular is outside the cell, digestion is just breaking things down into smaller pieces. And so extracellular digestion happens outside of the organism, and once things are broken down, then they're absorbed across the cell membrane of the organism. And so like I said before, this is something that we'll see in fungi, in some prokaryotes. Now, ingestive heterotrophs, for example, animals and protists, and so remember, protists are groups of unicellular eukaryotic organisms. There's two types of ingestive heterotrophy. There's intracellular digestion or phagocytosis. So intra, remember, means within. So within the cell digestion. And this is also called phagocytosis. And this is something that we see in protists, and we also see it in sponges, which are a sort of simple type of animal. The other type of ingestive heterotrophy that we can see is extracellular digestion. And that typically happens inside animal digestive tracts. So basically, ingestive heterotrophs, what they're doing is they're bringing something into either the cell or the body of the organism before they break it down into smaller parts that can readily be absorbed across cell membranes easily. So for example, in phagocytosis, there's a food particle that's not totally broken down to a small enough size to bring across a cell membrane. And then it's brought into the organism and then broken down within the organism, and then the food molecules are moved across the cell membrane. Or, as we see in animals, this process of extracellular digestion that happens outside of the cells. Basically, an animal will ingest food, chew it up, and break it down into chunks, and then it'll go into the digestive tract and be broken down even further so it can eventually be absorbed across cell membranes, and then those individual sort of smaller molecules can be used for cellular level processes. So just to sort of simplify a little bit of this, absorptive heterotrophs break things down, then take them into the cells, whereas ingestive heterotrophs take them into the cells or take them into the body and then break them down to move across cell membranes. Okay, so now let's look at the fungi, which are in a group or an example of an absorptive heterotroph. So remember that fungi spend the vast majority of their life as mycelium underground. So the mushroom that you see is just a short fruiting body life stage that produces spores for reproduction and things like that. But fungi spend most of their life underground as these sort of mycelium life stage that are a bunch of spaced out hyphae. And these hyphae have a very, very high surface area to volume ratio that's ideal for nutrient absorption. So if we were to sort of zoom in on these hyphae, they might look more realistically something like this, where you can see all these tiny, tiny little hairs that make up the mycelium. And this is really the feeding life stage of the fungus. And this filamentous structure, as I said before, it creates a really high surface area to volume ratio for digestion or breaking down of molecules outside of the mycelium And then once those molecules are broken down, they can be absorbed across the cell walls and the cell membranes of the mycelium into the actual body of the fungus, okay? And as it turns out, fungi actually have some of the highest or the highest surface area to volume ratio of any organism. So just for example, a cubic inch of soil, so just think about that, 1 inch as a cube, can have up to 1 mile of mycelium from one fungus threaded through it if you sort of broke it all down and lined the mycelium up end to end. So extremely high surface area to volume ratio. And so you can think of this surface area to volume ratio and put this in the context of structure and function, that idea of structure relating to function, and also this is a trade-off. So structure to function in the sense that having this high surface area to volume ratio really enhances the ability of fungi to absorb nutrients, okay? And so as an absorptive heterotroph, this high surface area to volume ratio is very useful. That structure of having that high surface area to volume ratio is related to that function of having to absorb lots of nutrients. But there's a trade-off here because having that really high surface area to volume ratio also means that fungi are very susceptible to losing water to the environment, okay? And so that's part of the reason that if you think about it, you don't really often see fungi in a desert, right? You think of fungi and molds growing in really moist areas and moist habitats. And so most fungi, because they have this very high surface area to volume ratio, are limited to habitats that have loss of moisture. So there's this trade-off there for the fungi. Okay, so go ahead and answer this question. All right, so if you said C, smaller single-celled prokaryotes take in nutrients less efficiently than larger unicellular eukaryotes because smaller organisms have a lower surface area to volume ratio than larger organisms. That is, in fact, the false statement. So remember that smaller organisms actually have a much higher surface area to volume ratio than larger organisms. And that's because surface area increases at a lower rate than volume does as organisms get larger and larger and larger, okay? All the rest of these statements are, in fact, true statements. Okay, so how do fungi get high energy carbon molecules and other essential nutrients? So remember that fungi, to eat, they need to absorb the nutrients that they eat through their cell walls and through their cell membranes. And what they're trying to absorb are reduced carbon molecules. Okay, so when we talk about reduced carbon molecules, we're talking about high energy carbon molecules because they need to get their energy and they need to get a carbon source from these high energy carbon molecules, okay? Because remember, they're chemoheterotrophs. And there's 3 major sort of strategies that fungi have evolved. They can absorb these molecules from dead organisms, and this is called being saprophytic. So saprophytic fungi will eat dead organisms, and those organisms can be, animals, they can be insects, or they oftentimes are wood or leaves or plants of some sort. They can also get these high energy carbon molecules from living organisms as mutualists. And so mycorrhizal fungi was the example that we used when we talked about mutualisms in plants, where the plants give sugar to these fungi that you can see growing off the roots, and the fungi in exchange absorb nitrogen and phosphorus that they give to the plant. So they're getting high energy sugar carbon molecules from these plants in this mutualistic relationship where both the plant and the fungus benefit. Or they can get it from living organisms as parasites. And so a parasitic relationship is a symbiotic relationship where one individual in the symbiotic relationship benefits where the other has a negative or a cost. And so Huitlacoche, this is also called corn smut or corn mushroom or corn fungus. This is a mushroom that grows on corn and has a very negative effect on the corn, but that mushroom gets lots of starch from the corn. And you can actually buy this canned to eat, and it's really pretty tasty. But this is an example of a parasitic relationship where one organism benefits, where the other organism has a negative effect. And just to remember, the sort of high energy molecules in these cases, that all of these cases that the organisms are getting, are often cellulose and lignin or starch, okay? So for example, starch is what's in corn. And remember, starch is a very complex carbohydrate. It's a polysaccharide. And so it's a bunch of these sort of glucose molecules that are linked together in these chains. And those starch molecules are really big, and fungi need to break them down before they can absorb it. Same deal with cellulose. So if you look at the cellulose that makes up like a dead log in the plant cell wall, you can look at that cellulose more closely. You can break it down, and what it is are these long organized chains of glucose that are very, very sort of strong and tough to break down. And so before cellulose, and lignin is a sort of similar structure that's part of plant cell walls. Before that stuff can get absorbed, it needs to be broken down into individual molecules. So just remember, these are complex molecules that have to be broken down before fungi can absorb them, okay? So how do fungi eat these big reduced carbon molecules like cellulose and lignin? Remember, they don't have a mouth, they don't have a digestive tract, and they also have cell walls that prevent phagocytosis, or that sort of pinching off of a food particle. So if you were to zoom in on the hyphae that make up the mycelium, you'd see that these are these sort of chains of individual cells that make up the hyphae. It's like a sort of system of pipes, okay? And so because they don't have a mouth or digestive tract and they have these cell walls, it means the food that they take in has to be small enough to pass through the membranes of these hyphae cells. And they have to do that either through diffusion, or they have to use protein channels that are embedded in the membranes that allow larger things to pass, okay? And so what that means is molecules need to be very, very small to move across a typical cell membrane. If we're ignoring the protein channel part and we just look at the sort of permeability, so we're going from low permeability to high permeability of a cell membrane, you can see that smaller nonpolar molecules like oxygen, CO2, and N2 can move very easily across a cell membrane. But as you get into larger polar molecules or some small polar molecules, remember these have a little bit of charge on them, and things like ions, it gets harder and harder and harder for these things to move across a typical cell membrane. And if you think about things like lignin, cellulose, starch, those extremely large molecules that we talked about in the previous slide, those things are going to be down here. It's very, very difficult for them to move across a cell membrane because they're so large, and also they can be polarized as well. Okay, so it's very difficult for these molecules to move across a cell membrane. So that means they need to be broken down into smaller molecules before they can move across. Okay, so that is this process of extracellular digestion, and this is what fungi use. So extracellular digestion means digestion outside of the cell. Okay, and in this case, food is digested outside of the organism. And it's the fungal hyphae that will actually secrete digestive enzymes that help break down these complex molecules outside of the fungal cells. And then once they're broken down, they'll absorb them. So for example, you can look at a complex cellulose molecule that's very difficult to break down, that's sort of these chains of glucose molecules all linked together. So the fungi will secrete some enzymes that will break the cellulose polymer into the glucose monomers. And then those glucose monomers are small enough that they can be absorbed across the cell walls and cell membranes of the hyphae into the body of the fungus. And you can see this log sort of being broken down here. So in other words, the resulting monomers and other small molecules that are broken down by these enzymes can enter the cells of the hyphae through this process of diffusion, or sometimes the hyphae will also use facilitated transport or active transport. And so this is just using proteins that are embedded, these sort of channels that are embedded in the cell membranes, that will be a more energetically expensive processes to sort of pump things against a concentration gradient. And this really depends on the molecule being absorbed. The big thing to sort of take home here is that because they are absorptive heterotrophs, they have this extracellular digestion to break these smaller, bigger molecules down into smaller bite-sized chunks that can be absorbed across the cell membrane. Okay, so use this text box to answer this question. Why do heterotrophs need to break down complex molecules like cellulose, lignin, starch, etc. Into smaller molecules in order to be able to eat them? All right, so remember, it's all because they have to move across the cell membranes. These things have to be broken down small enough so that they can easily move across the cell membrane. So you can see like a complex molecule like cellulose is just way too big to move across. But if you can break it down into something smaller like a glucose, it can move across more easily. You might be able to break it down even more, or you might be able to have some protein channels here to help these molecules move across more quickly. But ultimately, they have to be broken down so they can move across cell membranes, and those molecules can be used for cellular level processes. Right, so lignin, cellulose, proteins, etc. are all sort of down here and very difficult to absorb. Okay, next question. All right, if you put A, the rates at which fungi break down dead plants would decrease because their ratio of surface area to volume would decrease. This is the correct answer. So remember, if you increase thickness, essentially what you're doing is getting bigger and bigger and bigger. If you're getting bigger and bigger and bigger, your volume is growing at a greater rate than your surface area is, so your surface area to volume ratio is going down. If your surface area to volume ratio is going down, your ability to break down and absorb dead plants is going to decrease because you have a lower surface area to volume ratio. Okay, the last major point that I want to talk about is carbon cycling and saprophytic fungi. Okay, so remember, saprophytic fungi are those fungi that break down things like lignin and cellulose in dead organisms. And specifically, I want to talk about them breaking down lignin and cellulose from dead trees. So let's ignore the fungi for a minute and just think about trees, okay? So trees, remember, are photosynthetic, and part of that photosynthetic process is taking carbon out of the atmosphere and incorporating it into carbon in glucose that's sort of in the trees that the trees will use. So they need those sugars. And some of those carbon atoms will get used by the tree in respiration. So remember, respiration takes sugars and breaks them down and turns them back into CO2. And so some of those carbon atoms in red get turned into sugars, and some of those sugars, because remember, trees also do cellular respiration, will turn those carbon atoms and the sugars back into CO2 that will be released back into the atmosphere. However, a whole lot of that carbon that's absorbed initially by photosynthesis is going to get trapped inside the dead tree as lignin and cellulose, okay? So remember, we talked about this other process of biosynthesis, where some of that carbon that's incorporated into those sugar molecules by photosynthesis is also taken by this process of biosynthesis and can be turned into things like cellulose and lignin that make up the sort of trunk of that tree, right? And so that cellulose and lignin, these are notoriously difficult molecules to break down, and there's very, very few organisms in nature that have the ability to break down cellulose and lignin. Like humans, for example, you can't go just eat a tree or a branch and break it down. You need to have special either bacteria or fungi that are able to do that. And so what that means is that cellulose and lignin can really build up in an ecosystem, all those sort of dead trees and leaves and branches. Now, interestingly, what fungi have the ability to do, saprophytic fungi especially, is they can actually have the ability to break down the lignin and cellulose to get to those reduced carbon atoms. So remember, when I talk about reduced carbon, I'm talking about high energy carbon. Specifically, I'm talking about those glucose monomers that make up these complex cellulose polymers. And so all of these things are made up of individual carbon atoms. And so because they're breaking them down, they can use those carbon atoms that they absorb from those fungi for several, or from those logs for several different things. So some of that carbon will become part of the fungi's biomass. So this is once again that process that we referred to as biosynthesis. So all this sort of orange stuff that you're seeing on this mushroom here growing on this log is made of carbon molecules that it's absorbed from or taken from those plants or those dead logs. Some of that carbon, however, will also be used for respiration. So remember that fungi, in their cells, they also do cellular respiration. And remember, cellular respiration is taking glucose molecules and turning the carbon that's in glucose molecules, so this reduced molecule, into this more oxidized CO2 molecule. And in that process, producing ATP that can be used for cellular level metabolic reactions. And so some of that carbon will go back out into the atmosphere. And so what you've got here is carbon cycling. So it's moving from the atmosphere into trees, back into the atmosphere. These trees are dying, and some of it's moving into fungi, and then from fungi back into the atmosphere. And so this is a carbon cycle. Okay. Which statement is true? If you said mycorrhizal fungi get glucose from living trees, that is the true statement. So fungi are not ingestive heterotrophs, they are absorptive heterotrophs. Animals are not absorptive heterotrophs, they are ingestive heterotrophs. They take stuff in before they break it down. Fungi break it down before they take it in. Fungi use cellular respiration to incorporate carbon into their biomass. That's not true. They use these sort of biosynthetic processes to incorporate carbon into biomass. Cellular respiration is actually going to take carbon out of their biomass and release it into the atmosphere as CO2, as we discussed in the previous slide. Saprophytic fungi break down cellulose from living trees. Saprophytic fungi do in fact break down cellulose, but they do that from dead trees. If they're taking it from a living tree, then by definition, they are parasitic fungi because they're having a negative effect on that tree if they're breaking down their cellulose. All right, last question. Okay, so A is the false statement here. So fungi must take in carbon to incorporate into proteins. That's true. Remember, amino acids make up proteins, and amino acids are composed of carbon. So all organisms need amino acids, including fungi. All organisms need proteins, including fungi. Proteins are made of those amino acids. Amino acids are made from carbon. They need to get that carbon from somewhere. They have to take in phosphorus to incorporate into nucleic acids. So phosphorus, remember, is a fundamental part of every single nucleotide that makes up nucleic acids. Fungi have nucleic acids, they have DNA and RNA like all other organisms, so they need to get that phosphorus from somewhere. This is the false statement. They must take in N to incorporate in polysaccharides. It's true that they have to take into N, but that N is not being included in polysaccharides. So if you look at polysaccharides, so this is an example of a polysaccharide or many sugars. In typical sugars, they don't have nitrogen in them. They're just made of carbons, oxygens, and hydrogens, like this glucose molecule here. So that is the false statement. Okay, so as a summary, we've begun discussing chemoheterotrophs. We've talked about ingestive versus absorptive chemoheterotrophs, these two major strategies that heterotrophs use. We've talked about fungi as a great example of an absorptive heterotroph and how they have a very high surface area to volume ratio to break down and absorb complex polysaccharides that are way too big to get across cell membranes without being broken down. And we talked about the important role that fungi play in carbon cycling, and especially in breaking down complex, very difficult to break down molecules like cellulose and lignin that get captured in trees and plants.